A world of microbes. Science has probed it and revealed so many things about the tubercle bacillus, its body structure, how it functions. And now the electronic microscope is making its contribution to advanced research. Here is a tubercle bacillus magnified 12,000 times. But in spite of the vast results achieved, there are so many things to be studied. The study of tuberculosis germs has made steady progress since the time when it became possible to cultivate and propagate them. To cultivate bacteria, a culture medium must first be prepared. This is properly nourished with egg, serum, and various chemical compounds. Cultivation of bacteria requires a certain constant temperature in addition to nutrients. An incubator. The temperature within is maintained at around 37 degrees centigrade. Tubercle bacilli inoculated into the tubes of culture media are cultivated here. Some of the culture media contain dye called malachite green, which prevents the growth of other kinds of bacteria. These are the tubes of solid culture medium, made up of nutritive substance in solid form. Here are the tubes of liquid culture medium. This shows that live tubercle bacilli are within reach of the scientists at any time for study from all angles. A very fine glass tube, 1/100th of a millimeter in diameter, is now in the making. The glass tube is attached to a micromanipulator, a device for singling out one germ. Through this device, one bacillus is obtained to examine its characteristics. A tiny drop is drawn out of a liquid culture. This contains innumerable tubercle bacilli. By manipulating the glass tube under a microscope, a single tubercle bacillus is extracted. Here is the glass tube which has absorbed the fluid containing bacilli. The tube is extended over a solid culture medium on which is blown out a single tubercle bacillus. A tubercle bacillus, a microorganism 1/500th of a millimeter in length. An observation on the growth and propagation of a tubercle bacillus begins using a time-lapse camera. This high-precision instrument takes pictures at regular intervals by automatic flashlights. Tubercle bacilli are highly susceptible to the influences of heat and light. Therefore, a color which is the least detrimental to the growth of the germs is used for illumination, and when filming the temperature is also regulated. The microscope is set at 1000 magnifications. This is the virulent tubercle bacillus that attacks the human body. In the bacterial cell are tiny granules. these are seen squirming vigorously. When the growth of the bacilli stops, the movement of the granules slackens. These are the bacilli whose growth has ceased, and also the movements of the granules. These are growing, and the granules are active. We can see that changes take place between the granules, which result in the propagations of the bacilli. It seems that the granules have an important bearing on the lives of the bacilli. Let us observe the bacilli by photographing at a rate of 1 frame per 3 minutes. The bacilli propagate at a rate of once in 5 to 10 hours, far slower than the ordinary kinds of bacteria. How will the mass of the bacilli spread out? The observation through consecutive motion pictures continues. A propagating mass of bacteria is called a colony. Gradually it grows and grows. The microscope in use is set at 1000 magnifications as before, and the camera is taking pictures at a rate of 1 frame every 3 minutes. But how do the bacilli split, and how does the colony expand? Consecutive observations through the medium of the film certainly give the scientist valuable clues to the nature of the bacilli. The germs spread out like cords, and this is a characteristic of the tubercle bacillus, which afflicts the human body. The colony expands vigorously. Of course, we cannot say that the tubercle bacilli propagate themselves in this manner within the human body, but here on the culture medium, what had started from a single germ is seen multiplying into a huge, swelling and overflowing mass. It is a difficult task to distinguish the dead and live bacilli, but researches now in progress will make the task easier. The tubercle bacilli stain well with malachite green. If a red dye, phenylfuchsine, is added, it stains what may be presumed as dead bacilli. It seems that the contents of the bacterial cell of the dead bacilli are destroyed with the lapse of time, and the red dye seeps into the hollows. The bacilli in a vigorously growing colony with plenty of vitality remain green. They cannot be stained red. What happens if the virulent tubercle bacilli invade the human body? Since it is impossible to see directly into the body, the method employed today is to experiment with the germs outside of the body in order to obtain clues to the conditions within. A part of the spleen of a living animal is cut into a size smaller than 1 cubic millimeter, put onto a culture medium and kept alive in an incubator. Microscopic observation reveals that in 4 to 5 hours, wandering cells containing numerous leukocytes emerge one after another. Leukocytes are produced in various parts of the body and also enter into the blood vessels. Here we see leukocytes spreading out in all directions, like nebulae in the vast universe. The leukocytes cleanse the body of bacteria and foreign matter. These leukocytes are kept alive and observations are made as to how they trap tubercle bacilli and what becomes of those caught by them. Through such vital observations, scientists try to clarify the relations between leukocytes and tubercle bacilli. A single leukocyte seen through a microscope set at 400 magnifications. This is the most common type of leukocyte, called a polymorph leukocyte. It very much resembles the unicellular microbe called amoeba in shape and the way it moves about. The leukocyte also possesses granular objects which are seen in vigorous motion. Here we find tubercle bacilli and leukocytes placed on a special slide containing liquid culture medium. Now, let us see what happens. The leukocyte slithers up to the bacilli as if attracted by a magnet. It is presumed that the attraction is caused by some chemical reaction of the metabolic products of the bacilli. Leukocytes by nature try to engulf germs and digest them. The camera speed is 1 frame per 3 seconds. Therefore, the actual movement of the leukocyte is far slower. The leukocytes now gather and crowd upon the bacterium. We can imagine a similar occurrence in the human body invaded by bacilli. And so we know that the leukocytes are part of the defensive mechanism of the body. The monocyte, a kind of leukocyte. These increase when the illness becomes chronic. Granules are also found in them. They have thin, filmy cell membranes extending from their bodies. The monocyte is larger than the ordinary leukocyte and is slower in motion, but it has greater power over tubercle bacilli. How do these cells gobble up the germs? Now you can see for yourselves as the time-lapse camera depicts details of the monocytes in action. The microscope used is now adjusted to 400 magnifications as before. The camera speed is 1 frame every 10 seconds. The monocyte flaps its filmy membrane, writhes about and corners the bacterium. The white pine needle-like objects are tubercle bacilli. The monocyte gulps one germ after another quite greedily. Things like this are taking place every moment within the body. But how does the monocyte know that a germ is there? The monocyte shrinks and stretches its body as if aiming at the germ. Not so easy this time. Now will he get it? Yes, the germ is caught and engulfed by the monocyte. goes another. In order to observe the monocyte in action, let us cut the camera speed down to 1 frame every 30 seconds. What now? The body of the monocyte is shrinking and has become round in shape during a period of 6 hours. Now, here comes another monocyte. The camera speed has been restored to the former 1 frame every 10 seconds. The monocyte has already swallowed a number of germs, yet it goes on trying to gulp a mass of propagating bacteria. The granules are spreading out near the perimeter of the bacilli. How do granular things function? What mystery do they harbour? Scientists are intrigued by these questions. The mass of germs engulfed by the monocyte seem to be dismantled, taken to pieces. An astounding design, isn't it? The mass of germs caught and dismembered by the monocyte, will they be digested or will they survive? It is considered that some of them are digested over a long period, but as long as the balance of strength between the monocyte and the bacilli is maintained, the germs are believed to survive within the body of the engulfer. The observation continues. Why, the monocyte appears to be getting weaker. This is probably caused by some factors relating to the nutrients in the culture medium, or by some metabolic products of the bacteria it caught. At this juncture, a giant phagocyte shaped like a jellyfish has made its appearance, energetically flapping its filmy membrane. It approaches the weakened monocyte. The one on the left is a new arrival. The granules in this giant phagocyte are seen vividly. On the other hand, those in the weakened monocyte on the right are almost invisible. The jellyfish swims up closer. They have touched each other. Look what happens. The jellyfish stretches out its filmy membrane and catches the other. But at the same time, the jellyfish, with a show of fierce strength, has also engulfed the nearby mass of tubercle bacilli. The granules in the giant phagocyte are seen gathering around its victims. Now we can see that there is a cell which has the faculty of not only catching bacteria, but also of catching its fellow cells which have been prostrated. This incident, however, has suspended our observation to determine what will become of the germs caught by the monocyte. The suspended observation resumes with fresh subjects. The camera has continued photographing for about 70 hours at a rate of 1 frame per 10 seconds, with the microscope adjusted to 400 magnifications. In the center is a giant phagocyte of a chicken, which has already caught a number of germs and now is after more. These are avian-type tubercle bacilli which afflict chickens. Oh, the giant phagocyte is now trying to catch a small leukocyte just above it. But the leukocyte resists violently. While struggling with the leukocyte, the giant phagocyte continues catching the nearby germs. What will become of the tubercle bacilli already caught? Will they be digested? Will they survive? What relation can there be between the phagocyte and the tubercle bacilli? The struggle continues between the giant phagocyte harboring the bacilli and the small leukocyte. The leukocyte continues to resist violently against the phagocyte trying to devour it. This struggle has continued for 10 hours, when, all of a sudden, the giant phagocyte shrivels up, leaving behind a web-like trace. Yet it goes on living and moving. As also do the tubercle bacilli dotted around the two cells. At the upper right, pine needle-like bacilli are seen growing. What has now happened to the bacilli caught in the shrunken phagocyte? The phagocyte and the bacilli, they will keep on living as long as they can resist each other. But this balance of power may be broken. One hour has passed. Another. Still another. Bacilli in the field continue to grow, and the two cells their struggles. "22 hours have passed since we began our observation. 30 hours have elapsed. Gradually the bacilli cease growing, possibly for want of nutrition from the culture medium. But what has become of the bacilli contained in the phagocyte? "42 hours have passed. Suddenly a change, an explosive change, has taken place in the phagocyte. This change has lasted for 40 minutes, and the motion of the phagocyte is fast dwindling. As we continue watching and fearing it may die, suddenly the body of the phagocyte swells. Another explosive motion, and now the phagocyte is completely still. 2 hours had elapsed since the first explosion. The small leukocyte above is still alive and astir. Another 2 hours pass. Look, an astonishing phenomenon is taking place. A tubercle bacillus begins to emerge from the phagocyte, which is now dead. It is seen stretching out in the space between the dead phagocyte and the living leukocyte above. But it is fatter and longer than when caught. Still other germs are emerging from the dead cell below. The tubercle bacilli had stubbornly resisted death. and now they are making a vigorous exodus from the dead phagocyte. But the leukocyte is still alive and tries to catch them. However, the germs keep on propagating. At this moment, death came to the leukocyte, probably because the nutritive condition of the culture medium has deteriorated. While the cells die one after another, the germs keep on growing. We have seen how the tubercle bacilli remained alive in spite of being taken into the phagocytes. The long 70-hour observation is ended. Now we shall see the effect of chemicals on the tenacious tubercle bacilli. Let us give the bacilli a minimum of streptomycin, only 5 gamma or 1/200th milligram in 1 gram of water. The growing tubercle bacilli have been checked. The motions of the granules too are ceasing. And if hydrazide is given, 0.5 gamma or 1/10th of the amount of streptomycin is sufficient for inhibiting the growth of tubercle bacilli. Hydrazide is believed capable of arresting the growth of tubercle bacilli within the phagocytes and offers the most promising outlook for controlling tuberculosis. Yet there are so many things to be done in order to counter the inroads of this deadly germ of the white plague. By observing the interrelation of living cells and tubercle bacilli by means of time-lapse photography, we were able to ascertain how stubbornly the tuberculosis germ maintains life. All that takes place within the human body is highly complicated, and a number of problems are yet to be explored. But to reveal the world of the unknown, this is the task of science.